Hello everyone, good evening to all of you. Okay, so this is going to be the notes from my class that the professor went over immune disorders. And so let's start with a bit of his history slide. So we had this pair of scientists, I should say, Charles Richet and Paul Portier. And they hypothesized that the jellyfish or anemone stings had a toxin, and that was the reason why they hurt so badly. So in order to test this, they went and tested on some animals. Some of them did survive exposure. And then when they were given a second dose, unfortunately, the animal subjects died within minutes. The reaction, they theorize, was likely a destructive immune response, and this destructive immune response is called anaphylaxis. So this is a harmful or uncontrolled immune disorder response. And then they get the Nobel Prize for finding this out in 1913. Alright, so, immune disorders. First, we have hypersensitivity, and then we also have autoimmune, and then immunodeficiency. So in general, our first one, hypersensitivity again. I'll just be abbreviating it as HS for the purposes of this video, so we could get through them much quicker. This is the immune response. That results in tissue damage. So these, again, are mistaken identities, when the immune response mistakenly thinks something is wrong, and then you have tissue damage because of that in hypersensitivity. This is a sensitivity to the allergen, which is normally harmless. And this will involve the antibody IgE, that class of antibodies. Then we have our autoimmune. Auto again means self, so this is when the immune system actually just targets the tissues themselves. Again, autoimmune, that means self-immune disorder. Then we have the immunodeficiency. Immunodeficiency, whereas autoimmune is when the immune system is hyperactive towards itself, while hypersensitivity is overreaction to allergens. Immunodeficiency is when there's a lack of adequate immune response. So there's no immune reaction when there should be one. Alright, now let's talk about hypersensitivity. So we're going to go over the 4 types of hypersensitivity. The first type is, of course, type 1. And this is going to be involving those IgE responses. This is an immediate IgE-mediated response. So this usually happens in minutes with IgE, which is, again, a type of antibody. So the IgE will bind to the Fc portion to mast cells or basophils. So, of course, from our other immunology notes from my class that the professor went over last exam, we have the B cell with the allergen. And then this will differentiate And then this will form that specific IgE for that allergen. So again, the IgE, its Fc region, will bind to the mast cell, and this will function as the kind of like a captured antigen receptor. And when this happens, the mast cell that has histamines, so this will contain histamines. Again, it has those captured antigen receptors. So and then this allows the detection of invaders. When the antigen is bound, this releases kind of a bomb, because the IgE gets bound to something. So again, the antibody or the receptor is bound to something, an antigen or an allergen. This releases a bomb of these histamines. This process is called degranulation. And in degranulation, again, we're releasing all those histamines and inflammatory mediators. Alright, and during the second encounter, there's going to be some cross-links. And, of course, that's going to be, again, another degranulation process. There are a few types of allergens. We have inhaled, such as pollen. We have ingested, such as peanuts. Or we have injected, such as the venom from other animals. Alright, that's all on type 1. Let's go into type 1 immediate IgE mediated, which is still, or we are, sorry about that. We're going to be still in type 1. Got my notes a little bit mixed up. But since we still are in type 1, we're going to now be talking about localized and systemic anaphylaxis. So localized allergens. Include the formation of hives, which are going to have the wheel and flare. And then these will be blocked by antihistamines. We also have hay fever. Now, hay fever is basically an itching, which is also blocked by antihistamines too. Asthma. So when there's a respiratory allergy. And the inflammatory mediators are going to cause the bronchial tubes to spasm. And when these spasm, the mucus production gets thrown out of balance, and then antihistamines won't be effective on this situation. However, they will work on hay fever and hives. Now let's talk about systemic anaphylaxis, which we discussed earlier in the history portion. This is when the allergen enters the bloodstream. When the allergen enters the bloodstream, we get the blood vessel dilation, which will cause fluid loss. And then, of course, a drop in pressure. When that drop in pressure happens, this is what's called the anaphylactic shock. It's one of our terms, so I'm going to just highlight it. Again, anaphylactic shock for the systemic reaction. The bronchial tubes will actually constrict. And this could lead to suffocation. Alright, and then bee stings and peanuts and penicillin are causes. Fun fact, my professor, he's actually allergic to penicillin, yet he's a microbiologist. That's his specialty, that's his doctorate. Which is pretty ironic and also interesting. How you control these allergic reactions, you could use an epinephrine pen or an EpiPen. Again, epinephrine is another name for adrenaline, so you're triggering the release of that hormone and it's going to stimulate a response. Okay, let's go into type 2. This is a whole new type of hypersensitivity, so we're going to use a different color. Type 2 will be cytotoxic. This is going to destroy cells. Again, cytotoxic, that's going to kill cells. Alright, so the antibodies are going to bind to molecules on a cell surface. And this is going to actually cause the classic complement pathway. That's the one where we have C1 through C9, and then when we get to C3 convertase, we get this C3 via 3 pathways. We have the lectic, we have the classic, and then we also have the one that involves mannose, I believe. Or am I getting them mixed up again? Oh, no, alternate, the mannose/lactic, and then we have classic. So on any of those 3, we'll get to the C3 convertase, and then when we get here, again, we have that opsonization, or we have the triggering of the creation of the MAC, or the membrane attack complexes. We also have another outcome, but I can't remember it at the moment. However, when we're talking about the type 2 hypersensitivity, we have the classic complement pathway, and then this will lyse or do lysis via the membrane attack complex, or the MAC. This is what's going to create pores and poke into that cell that the body recognizes as harmful. The type 2 hypersensitivity could also trigger the antibody-dependent cellular cytotoxicity, or the ADCC. Sounds like a metal band or a rock band, I know. This will lead the natural killer cells, the NK cells, to bind to the FC regions of the antibodies, and then they're going to deliver the chemicals to kill the cell. So I'm just going to highlight the abbreviations. Some examples of this would be blood transfusions, if you don't match up the correct type or the correct donor with the correct recipient, you'll get the red blood cell lysis. It's kind of the main one, so we're going to highlight it. There's also hemolytic newborn diseases, as well as some autoimmune diseases as well. Alright, let's go to the next type. This is type 3 immune complex. That's the way it's going to be mediated, the immune complex. Switch to a different color. Okay, so the immune complex means the IgM as well as the IgG. And these are going to bind to soluble antigens. And then phagocytes are going to then bind to the FC regions. And then this will, of course, cause removal. If the antigen is in slight excess, smaller complexes form. This will trigger the blood clotting cascade. When this happens, we're going to also activate the complement system. This will call those phagocytes. And then, of course, the phagocytes are going to release those inflammatory cytokines. And then other toxic molecules as well. And this could occur during bacterial, viral, or protozoan infections. And then you'll, of course, have rashes, joint pains, farmer's lung, systemic lupus, erythemolysis, bacterial endocarditis. So we're just going to list some of these. So joint, rashes, farmer's lung, lupus, and endocarditis, bacterial endocarditis. Okay, we're also going to have something happen called the disseminated intravascular coagulation. And this is when there's the blood clot cascade and then the complement system. When this happens, the clots are going to form, and then that's what's going to cause the vital organs to fail. Again, there's going to be coagulation in the blood vessels. We also have the Arthus reaction. The Arthus reaction is the localized immune complex reaction. We're going to get to the systemic one in a bit. So after the injection of the antigen into a previously immunized individual, this is when injection happens too much. So you already are immunized and then you get another injection. This is what's going to happen, that Arthus reaction. An example would be when the tetanus diphtheria booster is given too often. Overuse, again, not taking the vaccine, but the overuse of it, taking it multiple times even though you already got that specific dosage that you need. Again, following dosage and all those, staying up to date on all your vaccines is good, but you don't want to, let's say, take this specific one 5 or 10 times. No, you don't want to do that unless it's required. Overuse of the tetanus diphtheria booster. Then we also have serum sickness. This is the systemic immune complex reaction. Right, I'm just going to, there we go. Okay, so this is going to be passive immunization. The antibodies provided for protection, or from the, are from horses and other sources like that, but these also have the potential to be attacked instead of being used. So they're going to be rejected by the body instead of being used. Okay, now type 4. This is the last type of hypersensitivity. Let me get to it here. This is delayed type cell-mediated. And looks like I was doing it in red. There's a delay because of those antigen-specific T cell responses. All right, so an example of this would be poison ivy. There's going to be that delay again of 2 or 3 days following the exposure. This could happen anywhere in the body. There's cell-mediated immunity, or cell-mediated adaptive immunity, I should say. So this is, again, another callback to a previous set of notes. Remember, the adaptive immunity is highly specific, so we're going to be using T cells a lot. This is going to be playing a central role in combating intracellular microbial infections. So, intracellular infections, and the effector TC cells will attack the infected host cells. This will damage tissue, though, since this is an incorrect detection in some cases. Right, so that was type 4. We also have different tests and other means of talking about the delayed type. So, first thing is the tuberculin, not tuberculin, sorry about that. This is a skin test. This is going to find the latent mycobacterium that causes tuberculosis. And the mycobacterial proteins are injected. And then this will create a thickened and a reddened site at where the injection happened. The effector TH cells then find the antigens, and then they're going to release the inflammatory cytokines. If the welt is big, that means this antigen has been seen before. It's another note to keep in mind. Alright, now let's talk about the contact hypersensitivities. So this is dermatitis. This is when the effector T cells respond to small molecules that penetrate the intact skin. These are also known as haptens. Again, from a previous lecture, the haptens will bind to the skin protein. Again, haptens are usually too small to elicit a response. However, this time we're binding with those skin proteins. And these are going to be taken to the dendritic cells. And then this is going to cause that presentation to the T cells. And when that happens, this causes rashes. Of course, there are a few sources of this: nickel jewelry, and then poison ivy is another good example. Also poison oak will be contact hypersensitivities and delayed tight cell mediated. Okay, now let's go into autoimmune diseases. Now let me switch this slide. There we go. So, autoimmune. Again, that means it will be fighting your own cells, the self. This is incorrect, of course. The immune system is very strong. We don't want it targeting our own stuff. So usually, lymphocytes develop that respond to self autoantigens. So autoantigens are antigens of the self, and usually these are taken out or gotten rid of. However, when we have autoimmune, this is the failure to remove these lymphocytes, these autoantigen lymphocytes. Some examples would be type 1 diabetes, which my sister was diagnosed with when she was in elementary school. This is mellitus. This is organ specific. This is where the cytotoxic T cells, so the TC cells, are going to be destroying the beta cells of the pancreas. And this prevents insulin from being made. There's also systemic lupus erythematosus, or SLE. And we're just going to use SLE, the abbreviation for it. This is, again, systemic. It's in the name. The antibodies are going to be fighting anything that basically has a nucleus. So the antibodies are against the nuclear material of own cells. So this could attack a variety of things, unfortunately. We also have the myasthenia gravis. Sorry again about all these pronunciation errors. In myasthenia gravis, this is systemic again. This is when the antibodies are going to bind to acetylcholine receptors, and this is going to block impulses. So no impulses. And then we also have the rheumatoid arthritis. Ailment. This is again systemic. The antibodies are going to be targeting the connective tissue, mostly in the joints. And this is, of course, going to be targeted by the T cells and the antibodies. We also have the neuromuscular autoimmune disease. This one would be multiple sclerosis. This is going to be a progressive disease, unfortunately. I'm going to just underline it. There we go. Okay, so this is when the TC cells attack the myelin sheath. The myelin sheath is going to be the insulation of the nerves. And when this happens, the neuron can no longer effectively conduct electrical signals, so this will stop neuron conduction. And over time, you're going to have impaired vision, speech, and neuromuscular function. And sometimes the cause could be a viral infection, unfortunately. All right. Now let's talk about immunodeficiency. Immunodeficiency, again, unlike autoimmune or hypersensitivity, immunodeficiency means that the immune system just doesn't have enough of a response when it needs to. So first we have our primary immunodeficiency. Okay, so the primary immunodeficiency would usually be congenital. And that word, when we use congenital, this means it's from a genetic defect. Or environmental factor. Okay, so we have antibody deficiencies. such as the selective IgA, selective IgA is when we see little or none produced. Selective IgA, there's also another type called a gamma globulinemia. So a gamma globulinemia. This is when few or no antibodies are produced. We also have lymphocyte deficiencies. One of them will be the severe combined immunodeficiency, which is also known as SCID. SCID is when there aren't any B or T cells in a great number or none at all. The bone marrow doesn't produce them. We also have the DeGeorge syndrome. This is when the thymus does not develop in the embryo. And then there aren't any T cells. We have a third kind here. There's also... Oh wait, no, not a third kind. Sorry about that. So, how do we treat these lymphocyte deficiencies? We could give a bone marrow transplant. That is one way for us to combat that. All right, now secondary immunodeficiency. So this is going to be acquired through means such as infection or stress. Should probably move this over here at the top. There we go. Alright, some viral infections actually deplete certain immune cells. Such as measles. These will replicate in the lymphoid cells. Then afterwards, they just destroy them. Another kind would be the HIV. And this will destroy helper T cells. And then, of course, HIV will lead to AIDS. We don't have those THs anymore. Other kinds are syphilis, leprosy, malaria. These are going to impact not only the T cell, but also the macrophage function. There's also multiple myeloma. Multiple myeloma is when there's a plasma cell that makes too many antibodies, and this will deplete the body's resources. Again, this will make an antibody that is made at the expense of others needed to fight infection. So when that isn't actually needed, there's a malignancy in that single plasma cell that proliferates out of control. Alright, now let's go and hit a pause here, and then in our next video about immune disorders we'll go through AIDS. But since it's a much different subject, I wanted to go and split this video off from here. Thanks so much for tuning in. Please have a good rest of the day. Do something nice for someone, and I'll see you soon.